Welcome to Pujari classes. Dear students, in previous class we have discussed about what is the structural organization of cell, how does the structure or the cell is organized, diffusion, osmosis and definition of the hypotonic solution, isotonic solution and the hypotonic solution. Dear students, now we have switched on to the topic, the another concept, cell. First one, what do you mean by the cell wall? It's the outermost covering of the plant cell. Cell wall is the outermost covering of a plant cell. In plants, in addition to the plasma membrane, have another rigid outer covering. It is called as a cell wall. Okay, what do you mean by the cell wall? In addition to the plasma membrane, the rigid outer covering. This is called as a cell wall. This structure is called as a cell wall. This is the plant cell. This is the cell wall, and the plant cell wall. It is lies outside the plasma membrane. This structure is called as a plasma membrane. The cell wall lies outside the plasma membrane. The cell wall is mainly composed of cellulose. It means the cell wall is made up of cellulose. Cell wall is made up of or composed of cellulose. Okay, what do you mean by this cellulose? Cellulose is a complex substance and provides a structural strength to the plant. Okay, what do you mean by the cellulose? Cellulose is a complex substance. Cellulose is a complex substance. And what is the function of cellulose? It provides the structural strength. Provides structural strength. It provides structural strength to plants. Okay, cellulose is a complex substance and provides structural strength to plant. This is about the cell wall. When living plant cell loses water through a osmosis, the cells, there is a shrinkage or contraction of the content of the cell wall. This phenomenon is called as a plasmolysis. Plasmolysis. What do you mean by the plasmolysis? Shrinkage or contraction of protoplasm or content of the cell. What do you mean by plasmolysis? Shrinkage or contraction of protoplasm or shrinkage or contraction of content of the cell. It is called plasmolysis. When it becomes the plasmolyzed, the cell when loses their water, that time the cell becomes the shrinkage. Okay, this is the plant cell. Plant cell. We got it dark. This is called as the nucleus. called as a vacuole. This is the cell wall. And this structure is called as a cytoplasm. This is the normal cell. When we put the normal cell in a concentrated sugar or solid solution, the protoplasm becomes the shrinkage or it becomes the plasmolysis. Put in a salt or sugar solution. Salt or sugar solution. So what happens? The cells becomes the shrinkage. Nucleus. This is the cytoplasm. This is the vacuole. Cell wall. This is called a plasmolyzed cell. Plasmolyzed cell. Okay, once again, what do you mean by the plasmolysis? The shrinkage or contraction of the protoplasm, it is called as the plasmolysis. When the cell loses their water through a osmosis. When we put the plant cell in the concentrated salt or sugar solution, the normal cells becomes a plasmolysis. Plasmolysis means a shrinkage of the protoplasm. Okay. Next, we conducted one activity. First, we know the requirements for this activity. Requirements. First one, Rioli or onion peel. Second one, watch glass. Third one, slide. Fourth one, cover slip. Fifth one, brush. Needle. Seventh one, concentrated sugar or salt solution. Next one, eighth one, water. These are the requirements for our activity. Okay. First one, reolene or permanent peel, watch glass, slide, cover slip, brush, needle, concentrated sugar solution or salt solution. Next one is the water. Then procedure. Mount a reolene in a water on a slide. Observe this slide under a high power of microscope, in a compound microscope. Note the small green granules called as a chloroplast. That chloroplast contains a small green substance that is called as a chlorophyll. Put a drop of a concentrated sugar solution or salt solution on a mounted slide and cover the chloroplast and observe the microscope. Similarly, we take another reolene, put in a boiling water for few minutes. The boiling water, it kills the cells. Then it is transferred into the slide and mount it. And again, first one observe, then again add the sugar or salt solution on mountain. Again observe it. Then what you observe from these slides? Okay, first one, this is slide. This slide is mounted by a normal reolene. This slide is mounted by a boiled reolene. This is a normal leaf. Normal leaf. This is a boiled leaf. Observe the microscope. Observation. Same procedure. This normally it is a placed in a sugar solution. Similarly, the boiled leaf is also placed in a sugar solution. And then after few minutes, both slides are observed. What to infer from this activity? To infer means what to conclude from this activity. The conclusion is what is your conclusion? First conclusion, the living cells are able to absorb water by osmosis. But boiled leaf, it kills the cells. The dead leaf unable to absorb the water. What do you mean by conclusion? The living cell. The living cell absorb water. The living cell absorb water by osmosis. Sorry, osmosis, not osmosis, by a diffusion. Correct. By osmosis. The living cell absorb water by osmosis. The dead cell. Dead cell unable to absorb water. Unable to absorb water. Okay, from this activity Only living cells shows their activity and interactions, but dead cells it do not show their activity and interactions. Cell walls permit the cells of plant, fungi and bacterial cells to withstand very dilute external media without bursting. In such media, the cells tend to take up water and the cells becomes the swelling. The cell wall it creates the pressure against. When water enter into the cell, the pressure creates in the cell. Automatically, the cell wall creates the pressure against the cell wall. So it increases the sizes. Okay, what do you mean? Because of the cell walls. This is the cell. When we put the cell in a hypotonic solution, hypotonic means a very diluted solution. When we put the cell in a hypotonic solution, the cells becomes the swelling. It does not bursting or does not blasting because of their cell wall. This cell wall creates an equal pressure against the swelling cells. So that's why it does not break. And also the cell wall is a resin and it is made up of the very complex substance. It is as the cellulose. Okay, dear students, in this class we discussed about the cellulose. Cell wall and also cellulose. The next, the features of the structural organization cell is a nucleus. We remember the temporary mounting of the onion peel. You also know a small dotted like structure or the peripheral region. It is called as a nucleus. You already know that who discovered the nucleus? The Robert Brown discovered the nucleus. Robert Brown discovered the nucleus. In an onion peel, we already did one activity. In that activity, the cells are rectangular in shape. In that cells, we observe a dotted line structure at the peripheral region. This is called as a nucleus. This nucleus is discovered by one scientist that is called as a Robert Brown. Now, I stop the lecture and continue the